Hello everyone, we might get started. I'm Emily Osier, I'm a member of IMAS's ResCom, and along with Nicole Hill, who's actually one of our presenters today, she and I host the MAS monthly Marine and Antarctic Study seminars, and it's our pleasure to do so. Today we're talking about marine biodiversity, and to someone who doesn't practice in that space, biodiversity essentially operates a little bit like an iconic term or a buzzword that's clearly extremely important, but the technical intricacies of it are far beyond the average lay person. Nonetheless, it's a term that's incredibly important for marine governance and decision making. So our three expert speakers here today are going to give us some varying views on where we're at now with understanding marine biodiversity in a technical sense, but also in a sense that makes it relevant for decision making. So the title of today's MAS seminar is Capturing Marine Biodiversity, New Tools and Glimpses of the Future. Our three presenters are Dr. Vanessa Lucia, followed by Dr. Nicole Hill, and then Professor Craig Johnson will wrap it up. I'll let each speaker introduce themselves briefly. Time is of the essence though, so we're going to have questions at the end of the three presentations. They've got a maximum of 15 minutes each, and I'm going to be a grumpy moderator. I'll be standing up and making very obvious body language cues if time runs over. Each of them will present from their fields of work on these latest insights and developments into marine biodiversity. So let's start off with a welcoming round of applause for this seminar and for Vanessa. Thanks.
Thank you. Thanks, Emily. My name is Vanessa. I'm a research scientist here at IMAS in the Centre for Ecology and Biodiversity, and I'm going to talk to you today about the future of quantitative seafloor analysis. So, the ocean represents around 71% of the Earth's surface, and since the first recorded measurements of bathymetry were made by British explorer Sir James Clark in 1840, acoustic remote sensing remains the most efficient method to map the seafloor. How effective acoustic remote sensing is for mapping the world's oceans is a function of ocean depth. This will affect both the swath coverage, the efficacy of the survey, and the resolution of the features that can be derived from that data. As it currently stands, around 7.3% of water depths in around 0 to 200 metres globally have been are represented by 7.3% of the ocean is in 0 to 200 metres. Has around 71% of this area uncharted. In the 0 to 200 metre depth range is the area most impacted by human exploitation and where the majority of our resources come from. In 2014, GEBCO created the General Bathymetric Chart of the Oceans, created the most recent bathymetric map of the world's oceans. They calculated that the survey coverage at around a 1 kilometre grid cell represents that around 18% of the global oceans have a single sampled depth measurement. That means that 82% of the global oceans are working on bathymetry that is interpolated or derived. Now, there are a multitude of reasons that we map the seafloor, and the bathymetry of the seafloor creates the foundation of a whole bunch of analysis, such as looking at biodiversity mapping, at research for looking at engineering, exploitation, for looking at submarine geohazards, for fisheries management, and for benthic habitat mapping. But today, of all the reasons that we talk about mapping the seafloor, we're going to talk about how we can use how we use bathymetry data to map marine biodiversity. Bathymetry data forms the foundation data set of biodiversity modelling, which Nicole is going to cover in the second part of this talk. And this is part of the habitat model here. From bathymetric data, we can derive a number of surfaces through geomorphometry, which can be used to describe benthic habitat distributions across the seafloor. When species are correlated with their observed habitat, we can create habitat models which describe the distribution of these species' habitats. If we can model changes in these habitats over time, we can predict future scenarios of changes in species distribution. The science of how we extract the attributes about the seafloor from bathymetry is called geomorphometry. It gathers together different mathematical, statistical and image processing techniques that can be used to quantify the morphological, the hydrological, ecological and other aspects of the seafloor. Today I'm going to talk about geomorphometry and its relationship with biodiversity mapping. In the past 8 years, the science of marine geomorphometry has undergone huge developments in both terrestrial and marine sciences, and this can be exemplified by the increase in terms in paper titles and conference series being held internationally. We can see here that since 1996, there's been a change of the terminology moving from terrain analysis to geomorphology. Technical advances in computing, analytical algorithms, and large data sets of topographical data have shifted the focus from digital representation of the terrain to more looking at process modelling and generalisation of these models. This was something that we were able to explore in a special issue which we hosted in 2007, where we collated together new trends and algorithms towards moving into quantitative studies of the seafloor and looking at the application of marine geomorphometry. Geomorphometry of the seabed not only determines the seabed depth, but it determines the topographic features in a top-down approach by using the acoustic remote sampling method. It allows us to inform the dimensionality of the ocean's shape, where that shape will affect the pattern and distribution of ocean currents, of circulation models, of sediment transport, of sediment layering. All of these processes link to the terrain of the seafloor at multiple scales. Seafloor geomorphology can then be directly linked to species distribution. But before variables can be derived from bathymetry, we need to confront a whole bunch of different processing challenges. Some of those challenges relate to the mechanisms that we use to collect the acoustic data, and for example, that might be spatial scale, where we're trying to combine data that might be collected from ships at one scale with AUVs close to the seabed at another scale. We have implications for that scale into the models that we use for creating the digital elevation model. There's also an issue in the fact that there's little to no validation due to the data paucity. In the beginning of the talk, I talked about the percentage of area that was actually mapped within different depth strata. Ocean exploration is still really new, and so our ability to actually collect data over time at multiple repeat locations is going to affect the uncertainties of how we can derive some of the models from these acoustic methods. Combining acoustic data from different frequencies, from different methods, from different offsets, and from different time periods, and different tidal corrections has an implication into the types of DEMs and the accuracy of the DEMs that we can generate. There's also implications that we have for data cleaning and removing the impact of acoustic artefacts. And we've done some studies in recent years to look at how we can use the DEM to look at quantitative methods of actually pulling out habitat. And here you can see the impact of an artefact here from the acoustics being translated through into the classification models. One of the other issues we have is the standardisation of mathematical algorithms behind the data processing. When we're looking at such features as creating slope of the seabed, we have examined how different algorithms for how you compute slope over different ranges of neighbourhoods using different software will have an implication on the results. Primary variables of marine geomorphometry always start with depth. From a review in 2018 of recent papers published on the topic, 100% of the analysts began with a slope derivation. The other most popular spatial derivatives were relative position, which refers to the location of the feature in relation to its geographic coordinate system, as opposed to absolute positioning. Planar and profile curvature, rugosity or ruggedness, and orientation or aspect were the next most popular variables used for describing the geomorphometry of the seabed. Slope, and this is rugosity. I should just mention this example is from the Huon Commonwealth Marine Protected Area or Australian Marine Park of the southeast of Tasmania. So we're looking at seamounts in this example. Once we have these multiple spatial layers that are derived from the bathymetry, we can use these layers to create rule sets around different classification models, such as benthic marine habitat or marine geomorphology. This example here is one of the models that we're looking at to develop geomorphological classifications of the seafloor, whether we can classify things into seamounts, into plateaus, into knolls or hills, canyons, valleys and terraces. We take the geomorphological classification of these shaped features of the seabed and we apply different rule sets to be able to extract the particular classes based upon the terrain attributes of slope and aspect and rugosity through building models. So using these models, we can apply different data sets at different spatial scales to understand how habitats and geomorphologies are spatially organised. These methods lead to quantitative estimates of spatial parameters, which we can then use to explain how seabeds differ from one another and may explain how habitats are distributed by depth and by latitude. And we can begin to do that quantitatively. We can then combine these results with biological survey data to create species prediction models, which can then aid in the management of marine reserves in this example. So what does geomorphological modelling mean for Australia? Around 15% of Australian shelf waters at 0 to 160 metres water depth have been mapped. Most of this data is represented by the Australian Hydrographic Office and the Royal Australian Navy, which are the largest holdings of this data, accounting for around 12% of the 15% of total area mapped. CSIRO has collated around 2% of this data within the 160 metre depth strata, and state mapping accounts for around 1%. In Tasmania, this example here shows that we've mapped around 16% of our state waters inside of 160 metres, with New South Wales having the largest area mapped. As we increase data holdings at spatial scales relevant to species distribution, our ability to improve the tools for biodiversity mapping is ever increasing. National efforts to coordinate surveys and bathymetry data have evolved just this year in 2018 through the initiation of AusSeabed. AusSeabed is a national seabed mapping coordination program aimed to serve the Australian community relying on seabed data by coordinating the collection of data surveys, up-and-coming surveys, and actually looking at national priorities around Australian waters. The program is a collaborative initiative which is being led by Geoscience Australia, but has representatives from each Commonwealth state and territory, from universities and from industry. In the spirit of collect once and use many times, this is the initiative that's taking us forward into being able to look at big data and the collation of a national approach. When we overlay ecological data, such as data that can be derived from CMAP Australia, another one of our platforms that we've developed here at IMAS, we are well on track to improving our understanding of how geomorphology influences the distribution of benthic habitats. It's only when we combine the ecological data at meaningful scales relevant to the geomorphology that we actually begin to understand benthic habitat distribution. Understanding the scales that bathymetric models and geomorphological variables are most relevant to ecology is probably one of the most significant challenges in this field at present. We are currently working towards the development of methods which will prepare us for species modelling at high resolution as biological data becomes available in the future, which is something that Craig will talk about in his talk. So the development of these national methods of linking and understanding the distribution of geomorphology and how we create those variables for geomorphological mapping and linking those to ecologically relevant scales is going to be really the way we move forward into the future. Seabed 2030 is a global project which is going to look at mapping the world's oceans at 100-metre resolution by the year 2030. Once these geomorphological models become available and we can begin to understand how the errors and uncertainties in creating the variables for geomorphological modelling can be adapted, we can begin to start looking at global species prediction modelling. I'll now pass you to Nicole to talk about the statistical tools for how we are going to move forward in this space. I'm a little short.
Hello, that's better. I'm a little short on breath today, so I'm going to use this microphone. Thanks, Ness, for setting the scene. So I'm a research fellow here at IMAS who has an interest in modelling biodiversity, and I work closely with statisticians and other ecologists from a range of different institutions. And despite that nasty word in the title, statistical, never fear, I don't actually think in equations, so there won't be any in this talk. So, understanding the spatial distribution of biodiversity at broad scales is needed for a range of management applications. These include prioritising conservation, and generally we're talking about some aspect of spatial conservation here, regulating human activities and understanding and helping to manage responses or potential responses to climate change. But the fact of the matter is that biological data is by its very nature hard to get, and it's sparse and it's patchy. So if we want to understand what's happening at broad and contiguous spatial scales, then we need some kind of modelling to be able to do that. And generally we're talking about modelling that links this biological data to environmental data, such as Vanessa just presented, that describes why species might live where they are, so what their habitat preferences are, but has full spatial coverage to allow us to generate generally maps about where we expect to find different sorts of biodiversity. So biodiversity, as Emily said, is potentially a buzzword and means lots of different things to lots of different people, and we could debate what we were talking about biodiversity all afternoon. But here, what we're talking about is essentially multiple species and assemblages when we're talking about biodiversity. And modelling this kind of multivariate data is actually quite challenging. So this is the type of data that we start out with. We have a number of species that we've observed and a number of sites that we've gone to survey. So this might be on a ship, this might be going out looking at rock platforms, whatever the system of interest that you're interested in. And the way that we would traditionally deal with this data as ecologists is that we would turn this site by species matrix into a dissimilarity matrix. And this dissimilarity matrix describes site by site how similar the species, how similar the assemblages are between pairs of sites. So it's one number that describes this, condenses all of that information about species into a dissimilarity site by site. So we get this matrix. And then we'd run a whole a range of dissimilarity metric approaches over it. These analysis approaches here that some of you might be familiar with, depending on our question. But once we've done that, we're talking about dissimilarities, and we need to then do another step to dig a little bit deeper into what's actually driving these patterns. So ultimately, we're actually interested in questions about which species are found where, what are they responding to, and how are they responding. But because we've turned our information about species into a dissimilarity matrix, we've actually lost this information, and we generally have to do something else to try and recover it at the end. And often, prediction is either very difficult or not possible with these types of methods, or interpreting what that prediction actually means is also problematic. But there is another way. In recent years, there have been some substantial advances in ecological statistical modelling. And I'm referring to these as multi-species model-based approaches. And essentially, what they are approaches that can model the occurrence or abundance of species directly. And they're based on, so under the hood, they're based on GLMs, multivariate versions of GLMs. And depending on the type of question that we're answering, they usually have some component of a latent variable. And this latent variable is essentially something that you haven't measured, but that can come out of the data. So we might be talking about things like trying to account for the co-occurrence or correlation of species that you see at a site. So obviously, if you go to one site and you measure 10 different species, they're not necessarily, whether you find them there is not necessarily independent of each other. Or you might be trying to find groups of species that are responding similarly to their environment. And the advantage of directly modelling the species is that we have a direct interpretation of what's actually going on in terms of which species are responding to their environment, how are they responding to their environment, and we can generate predictions in a fairly straightforward manner about species or groups or whatever we're actually interested in. So multi-species model-based approaches allow us to do a number of things. They allow us to quantify how species and biodiversity responds to environmental and other factors. And here's an example here from Scott Foster's work at CSIRO, where they looked at the cumulative impacts of fishing. So they were looking at whether there are groups of species that respond similarly to fishing pressure. And they found a whole range of different groups that responded in, 5 groups that responded in different ways. But within that, this is an example of one of those groups, and within this group here, there might be, I think there are like 10 different species or so that were responding in this general sort of way to increased fishing pressure. So initially negatively, then an increase as fishing pressure increases. We can then turn that information about the species and their responses to some environmental or human pressure into maps. So this is looking at how the catch of these species has changed with the cumulative fishing pressure that's been in this area since 1997, I think it was. But we also get information about the species, which species are in this group. So generally these were species with short generation times, short lifespans, low average age and maturity. So we get some ecological information that we can use to further interpret what's going on here, which is obviously quite useful. We can define and describe biological assemblages and map their spatial distribution. So I'm going to give you a quick example of this in the next few slides. We can detect, attribute and model ecological change. We can predict future change. And of course, all of these things have applications to spatial management and monitoring. But we can also gain some more insight ecologically as to what's happening as we're answering these type of management relevant questions as well. So we can model species co-occurrences, which gives us information about networks of species interaction. So it might be under different scenarios. So under some kind of natural scenario, these are the species that are interacting positively and negatively. If we apply some management action, this is what happens. We get a different pattern of species interactions. And we can look at the effects of functional traits. So do particular functional traits moderate responses to different factors, essentially. So this is an example of where we've used a model-based approach for eco-regionalisation. So the idea here is to model and map where assemblages are in space, and it's a method called the regions of common profile. So we've looked at this on the Kerguelen Plateau using demersal fish data. So normally if we were doing this type of analysis, it would be done in 2 stages. First, you'd have to either decide how many groups there were and then match those groups to their environment to model and map them, or you'd model each of the species individually, then get a stack of maps and then have to group those and somehow decide on that and interpret that. But this method actually does both of those steps within itself at once, which means that essentially not only would we get the distribution of our different groups, and here we've found 7 different groups or assemblages, but we also get some kind of uncertainty, and this kind of uncertainty is an appropriate kind of uncertainty because it's actually looking at what's happening in both of those stages, because it's not actually doing it in both stages, it's doing it in one stage. So our uncertainty is actually reflective of in our model. So if we dig a little bit deeper into this, what do we get? If we look at this yellow group here, we can actually get from the model directly what species are we likely to find there. So we've got a whole group of species which we're unlikely to find, some species which we're highly likely to find, and other species which have varying levels of likeliness to find there. So we get this profile directly from the model of which species are we likely to find, which are we not. And we can use that to define which are our characteristic species. And because we've got this profile with error bars around it, we can actually go back and plug new data in here and validate against this directly, see how good our model is doing. But the other part of the model that comes directly out of this is essentially the environmental component. So what are these groups responding to? In this case, the yellow group up here is a shallow group and has some relationship with chlorophyll A, so generally found with so generally more likely to be found where there's more chlorophyll A. And of course we can use these type of products for a range of things. So we've used this to look at the representation of these assemblages in the current Himi Marine Reserve, but you could also use this as a planning tool. Next example is looking at mapping the distribution of benthic biodiversity. So Ness was talking about getting better environmental variable layers that mean something for the biology. So this is the work of Jan Janssen in our lab, who has taken this well, not one step further, but from a different angle really, and has been looking at, one aspect of his PhD was looking at modelling the availability of food to seafloor communities, because obviously that's something that's going to drive where you find them. And has a rather neat little model for doing this that combines satellite data, oceanographic models, and so on. And I refer you to this paper to have a closer look. But once we have these type of environmental variables, we can then start to look at, and this was done in a region of East Antarctica that's currently being proposed as an MPA, at species that share similar environmental responses, and we can map where they are and what they're composed of. So in this particular analysis, I think Jan found 5 different groups. This particular group here is a group that's sort of dominated by sessile filter feeders and has quite a distinct distribution found on slope areas and high current areas, versus another group which has a slightly broader distribution found on banks and some of the areas with less high current flow, and has a greater range of species that characterise them. So this kind of information can help managers characterise what's actually in areas that they care about from the small amount of localised data that we actually have. And now we're at a stage where, excitingly, we can start to think about rolling this out around Antarctica by developing this model, combining it with imagery data we have all around Antarctica, and start to think about circumpolar distribution of ecoregions, and also potentially key groups that we might be interested in, like vulnerable marine taxa, which have consequences for the fishing industry. So we can also, where we have time series data for particular assemblages, we can also start to think about digging deeper into how assemblages have changed through time and why. So if we have time series data, not necessarily in the same place at the same time, and environmental data through time, and sometimes auxiliary information that we might be interested in, and run them through these types of models, we can understand how species respond to their environment, so which species are responding to temperature and so on, how the function affects these responses to their environment, and we also can generate maps of how the distribution of individual species, and any combination of that we like, have changed through time. So this gives us great power to sort of drill down into what's going on with assemblages and understanding change. And then once we have this kind of information about what's happening in recent times, we can then couple that with climate projections to try and understand how distributions of species might change in the future and what that might mean for the structure of assemblages. So another aspect that we can use this type of approach for, once we've got these maps about how species have or are likely to change through time, is that we can use them to identify key regions or potentially indicator species that have or are most likely to change now or into the future. And then we can use that as the basis to sort of hone or optimise monitoring designs. So we can then drill in down into our maps of the species we're interested in and say, if we place transects or samples here, here, here and here, and we've seen this change through time, or we expect to see this change through time, do we capture that in our sampling design? In this case, no, but if we put them somewhere else, we might. So this can help us optimise where and when and how often we should sample. And this is a big question at the moment in the Southern Ocean, trying to optimise resources to detect change into the future. So in summary, multi-species statistical models are a very flexible tool. They can provide us with new ecological insights. They have advantages over some of the other ways that we traditionally model multivariate data or multi-species data, including they have direct interpretation. The predictions are fairly straightforward. We can appropriately quantify uncertainty, and we've got the potential to validate. And all of these things come directly out of the one model without having to do some kind of auxiliary analysis. And of course, in addition to the ecological insights that we can gain from this, that has many applications. So we can inform spatial management of biodiversity in a number of ways. It can be used potentially as an analytical tool in and of itself for monitoring. And we can use the outputs of the mapped outputs that we generate as tools to help us inform biological monitoring and observations into the future. That's it. Thank you.
Is there a point on this?
Yeah.
Okay, thanks. Good afternoon. I'm Craig. I head up the Quality of Biodiversity Centre here at IMAS, and my normal state of health is normally much better than it is currently.
Sorry, Craig.
Oh, is it? Oh, okay. In terms of biodiversity data and information, I thought I'd just focus on one example, and that is eco-regionalisation. It is just one use of biodiversity information, but it's used for all sorts of things, particularly around really important decisions like marine spatial planning, policy development. I'm not going to go through the whole list. The reason I thought I'd pull this one out as an example is that in Australia, a lot of these decisions has been based on the so-called IMCRA version 4, the Integrated Marine and Coastal Regionalisation of Australia. This was released in 2006. It was based largely on demersal fish, a fair bit on geomorph and sediments, much less on there was some marine plant and invertebrate data, and for the offshore sites in particular, some oceanographic data. But a lot of the data behind this was only up until about the end of the 1990s. So, so that's now a long time ago. So what I want to really emphasise now is that the kind of data that are either available to us now or about to become available to us is far in excess of what we've got here. This is what IMCRA produced, these bioregions, and this map has driven a lot of very key decisions at government level. Most marine scientists would say that this map is rubbish. There's some elements of reality in this, but no one's going to hold their hand on their heart and say, yes, this is really the way that it is in terms of the way that our bioregions are distributed. And at least I don't know anybody that would hold their hand on their heart and say that. So with very imperfect information, we've been making some pretty important decisions, I guess, is the message. There's a lot more data that is now available. I hope all of you are aware of Reef Life Survey. These are the sites around Australia. You can see the coverage is fantastic. Fish, invertebrates, and from images from photoquadrats, algae data can be obtained. The data are open. You can the numbers in those circles represents the number of transects. So it's huge. These are the sites from Timahara at Museum Victoria. Each one of these dots here is a sampling site. These are ophiuroids, the brittle stars. Some of these data were used for IMCRA, but this data set now represents a phenomenal and valuable collection around which a lot of these kinds of analyses can be based. talking about images, these are locations around Australia where these baited remote underwater videos have been taken, and there's recently been a national effort to try and handle images in a very consistent way. Imagery is a really important source of data, and we're seeing an exponential increase in the nature of the data that can come from that. So that's the BRUVs. Speaking of images, all these sites here are from the autonomous underwater vehicle that the IMAS facility runs. A lot of these are in moderately deep water, and you can go to. It's possible to go and pull up any of that imagery. Then there's work currently underway to do automated analysis of that imagery, which will open the floodgates on the data run. I've got this slide from Nicole. I mean, it's a bit old now, 2013, but all of these pink crosses around Australia here are from the CSIRO deep water video, and there's a lot more pink crosses now if we could bring this up to speed to 2018. The message I'm trying to get across here is that we're on the verge, it's already started, of having massive amounts of open data available for biodiversity analysis. So that puts us in a tremendously powerful position. And Australia is ahead of most other countries. In fact, I can't think of anyone who's running a national effort any better than we're doing here in Australia in terms of collecting this kind of information and making it available in an open way. Vanessa mentioned CMAP Australia. It's a marvellous new piece of technology. Again, it really is a global first in providing a single national classification scheme for seabed habitats. But in addition to the habitat layer, there's 90-something other layers that are available. So you can make maps here in real time. Whatever you want to show on that map, you can produce that map. So this example from up here is we've turned on the marine habitats, we've turned on the Commonwealth Marine Reserves. This is the Montebello Reserve here. It tells you that 64% of it is in fact unmapped. We've switched on the oil and gas pipelines. All of these little black dots here are the wells, so you can basically pull up any sort of map that you want. And the response to CMAP Australia from many people in being able to do this has been really fantastic. But CMAP is not just about producing maps. CMAP is joined at the hip with AODN, which is now been around for a long time, and again really stands as a global gold standard of how to do this sort of thing. And from any of the data available through any of the information available through CMAP, you get a direct access to data and the metadata, so you know about data provenance. And because of the ISO standard behind that metadata, there is the capacity for interoperability of those data sets. And that straight away enables you to take on all this sorts of modelling, the kinds of stuff that Nicole has just been speaking about. So I want to go back to this slide that Nicole showed you just to emphasise a few points. So one of the critical points is that, yes, you get these hard classes here, and you get complete spatial coverage. No one anywhere is, of course, going to monitor the entirety of their continental shelf or whatever area. You get bits and pieces of information, and then using the modelling, you get the 100%, the 100% spatial coverage. But what's critical with that, in addition to the fact that there's sophisticated cross-validation in these sorts of techniques, is that you get this uncertainty map. And that helps enormously for people that then are going to go and make decisions on the basis of this kind of information here. decisions around policy, but also decisions about where you're going to next put your instruments into the water. So if I was looking at something like that, I'd see this region through here of low probability, and if we were heading off down there on a voyage, that would clearly be an area to target in terms of improving the information on that sort of thing. Again, the fact that you can get all this information, you've done the multivariate bit, and whoops, what's happened there? You've done the multivariate bit and produced produced these different hard classes, and you've recognised that in this particular class we've got these species with these sorts of abundances, and then relationships to environmental conditions. This kind of information, with the validation and with the uncertainty maps, is just orders of magnitude in front of where we've been for stuff like IMCRA. So we are really poised in Australia, and it's right at our fingertips now for us to be producing this kind of thing for the entire continental shelf of Australia. It will completely revolutionise, or it should completely revolutionise, decision-making in marine spatial planning, in policy development, and in all of those areas that are really important for ensuring ongoing stewardship of Australia's marine estate. There is just no comparison between this kind of approach and this kind of approach, and we are really at the point of being able to do that on the right-hand side. There's also other kinds of modelling that biodiversity data can enable, and I know this looks like a bit of shameless self-promotion, and it is. It's an example of some work that was done through the Super Science program that myself and Neil and Neville Barrett were leading, and some work from Martin, who was the postdoc on there. So the sort of thing we could do here, and these data came from the autonomous underwater vehicle, the IMAS underwater vehicle, focused on the deep reefs in 30 to 100 metre range, usually out of sight, out of mind. We can't dive and do meaningful work on those depths. It turns out that they're very rich in terms of biodiversity, so we should know a whole lot more about them. And this kind of modelling enables this kind of thing. So here we are in the 2010s, and we get an abundance of this particular stalked ascidian, which is here, in terms of the probability of its presence. So it doesn't occur down here; it's mostly in this region and up here. Now, because we have information about the environment there, we can model the relationship between the environment of this group, and then from Eric Oliver's projections of how that environment's going to change through the 2060s, we can then start saying, well, this is how we might anticipate in this particular species to respond to that change in environment. So there's the 2010s, there's the 2060s, and here's the change. So it's going to get a whole lot less abundant on this part of the coastline and a whole lot more abundant in this part of the coastline. And here's the 2010, and there's the 2060 with the change in latitude down the east coast. So we were able to do this for a whole bunch of different functional groups, and out of that came some information which I suppose is not too surprising, but certainly hadn't been done in this way previously, and that is that different functional groups respond differently to these environmental variables. So we don't expect, looking at these kinds of data, each of these different functional groups to just all move in the same way. You know, we know that everything's shifting south, but they're not going to shift south in the same way. So what that means, the logical conclusion of that, is that what you end up with in the 2060s is a different relative abundance of those different functional groups on these deep reefs. And because we're talking about functional groups, by inference, it means we're going to see a shift in ecosystem functioning along that kind of gradient as well. So this is just one example of that kind of modelling. A limitation of this sort of approach is that it's really doing it functional group by functional group, and we're not taking into consideration the fact that a shift in relative abundance of one functional group might influence the abundance of another one that it interacts with. And that brings me to this example, which comes from the last chapter of Jan's PhD thesis, where we were really here trying to get him to look at both the prediction in space and to see how we might make some predictions about the dynamics of communities. So again, this is based on the work in East Antarctica. So having produced the map of the distribution of the species of communities, with that map and knowing other knowledge of the system, to build basically a qualitative network diagram of how those different functional groups would interact with each other and what the key drivers were, which can be some physical ones. We can then press those key drivers, say, okay, make it, so if a key driver is depth, we could make it deeper. And then we should see, if we make it deeper, a shift in the relative abundances of these functional groups, which we can then test back onto this. But then we can start taking some of the biological things as well and say, well, what happens when we press those? So this is that network. I'm not going to go through the detail, but you can see here slope of the seafloor, which affects the hard substratum. It's going to affect food deposition and sedimentation rates, depth of the seafloor, pelagic food sources. This here really refers to the primary production up on the surface, the phytoplankton production. So what was really useful with this kind of approach in taking this kind of biodiversity data is that by changing the slope of the seafloor and the depth, we were able to see how all of these things here would change according to this particular model. Check to see that that's in fact what we were seeing in the real world, and it turns out that it was, which gives Jan some confidence that he's actually got that network model correct. And then we can say, well, okay, what happens into the future when we know we're going to get more phytoplankton in this area because we're going to lose the ice? And he can run that analysis on that network. So for the first time then, we're in this position where we can, it's not just describing what we've got on the bottom right now, but it's looking at prediction around the dynamics into the future, which is incredibly useful. So just to sum all this up, what have we got here? Really on the edge of major changes in the amount of observation that is happening, and that these data are made as open data through these kinds of technologies here. Interesting that AODN, CMAP uses a lot of data from AODN, but then we serve data products back to AODN. But once you've got that sort of thing, then the modelling is possible. You can do the biodiversity prediction in space and time, the community dynamics ahead, and then that enables you to have better policy management and stewardship. That's the environment in which we're headed now. I have to say that, again, I'll say that Australia, I think, is ahead of the rest of the world in this kind of big picture stuff. This is the team across IMAS and CSIRO and the AODN folk that are really delivering something that's quite special in the space. In terms of IMAS, Nicole and I had this quick mind mapping exercise around what we were doing with the other folk at CSIRO involved as well in marine biodiversity. And these are all the kinds of impacts that we're already having from this sort of work. Most of these are current end users. Some of them are ones that we're expecting in the future. There's a bunch of current projects here. These collaborators, international collaborators here, are all part of existing grant applications. So there's a lot of activity in this space, basically, is the message that I'm trying to get across from this. It's a brave, exciting new space to be working in. And I feel really proud of the Hobart community that's involved in this stuff, in really taking this to the rest of the world and leading the thinking in this space. Thank you.